A signal transduction pathway is a series of steps through which a signal received on a cell's surface is converted into a specific cellular response. One type of membrane receptor is the G-protein coupled receptor embedded in the plasma membrane. It acts as an on-off molecular switch depending on whether GDP or GTP is attached to the G protein. When GDP is attached to the G protein, it's inactive, but when GTP is attached, the protein becomes active. When the specialized signaling molecule or ligand bonds to the G protein coupled receptor, the receptor is activated and changes shape. The inactive G protein then binds to the receptor and is activated when a GTP replaces the GDP. Once the G protein is activated, it separates from the receptor and diffuses along the membrane and binds to a specific enzyme. The enzyme is activated and changes shape. After the enzyme has been activated, it can trigger the next step in the signal transduction pathway, which goes from relay molecule to relay molecule until the cellular response has been activated.
In this case, the cellular response is the release of calcium ions from the ER.
Tyrosine kinase receptors are plasma membrane receptors that attach phosphates to tyrosines. Because one receptor, tyrosine kinase complex, can activate many different transduction pathways and cell responses. It can help the cell regulate and control many different aspects of cell growth and reproduction. This is a key difference from the G protein coupled receptors, as the G protein coupled receptors cannot trigger so many different transduction pathways. Prior to being activated by a ligand, the tyrosine kinases exist as inactive monomers in the cell membrane, where each monomer has an extracellular ligand binding site, an alpha helix in the membrane, and an intracellular tail containing multiple tyrosines. When a ligand activates the receptor kinases, the two monomers move closer together to form a dimer in a process called dimerization. The dimerization of the tyrosine kinases activate the tyrosine kinases region of each polypeptide chain, and each tyrosine kinase adds a phosphate from an ATP molecule to a tyrosine on the tail of the other polypeptide. When the protein is fully activated, it can be recognized by specific relay proteins inside the cell. Each individual protein binds to a single phosphorylated tyrosine and undergoes a structural change that activates the bound protein. Each of the activated protein triggers its own transduction pathway that leads to a cell response.
In this case, the cellular response is the release of calcium ions from the ER.
Type 2 diabetes is the most common form of diabetes. People with type 2 diabetes can make insulin unlike those with type 1 diabetes, but their bodies cannot use the insulin well or do not make enough. Because the insulin is not used as it is supposed to be, glucose builds up in the blood. In type 2 diabetes, the cell sends insulin signals to the cell, but the cell has lost its ability to respond to those signals. This can occur over a period of time in people who are overweight or physically inactive. The signaling pathway is not disrupted when insulin bonds to a tyrosine kinase. It's just that the cell pathway does not respond because the signal is too weak. Phosphorylation then does not occur. This is because low levels of the protein beta-ARR2, which regulates the sensitivity of insulin signals, is not present as it should be, therefore causing the pathway not to occur. The symptoms of type 2 diabetes are fatigue, blurred vision, weight loss, increased thirst, and frequent urination. Currently, there is no cure for type 2 diabetes, but it can be managed by eating healthily and exercising while maintaining a healthy weight. A person can also take drugs that decrease the glucose production and increase insulin sensitivity, lowering the amount of sugar in the blood.